we are very, very glad to welcome tonight Judy Lieberman, who is an MD, PhD, and she holds an endowed chair in cellular and molecular medicine at Boston Children's Hospital, and she is a professor of pediatrics at Harvard Medical School. So Judy has an extraordinary career, and I was not sure where to begin. Because, well, she published more than 250 papers in the most prestigious journals. She did very important contributions, and to choose one was impossible. Also, she received many awards. Well, I will just mention a few discoveries. But first, let me tell you a little bit about her unusual background. Judy was not a biologist from the start. After attending Rockefeller University, she first received a doctorate in theoretical physics. And during her first postdoc, she studied general relativity and quantum field theory. So, and I mean, she published papers on these topics. But sorry, I have not read them. So, well, so after about 3 years, she decided to leave physics and she go to medical school. So maybe, I don't know what was the reason, but she go to medical school and she got her MD from a Harvard MIT program in 81. So she was in 1981, so she was get into the field of HIV since the start, being a resident in internal medicine. She then did a postdoc in the MIT Center for Cancer Research Laboratory at Hermann Eisen, with Hermann Eisen. And little by little, she became both an immunologist and an instructor in medicine. In 1995, she moved to Boston Children's Hospital, where she became a full professor in 2004. And, well, to mention one of many of her responsibilities, because there were really many responsibilities, she served as the director of the Division of AIDS at Harvard during about 5 years between 2005 and 2009. So this was a really very quick overview. There are many, many other things to say. Just let me tell you a little bit about her major achievements. So, she contributed to the field of immunotherapy since the beginning, and she can witness how AIDS research shaped the study of immunology. She designed and performed the first clinical studies of adoptive T-cell therapy, and she discovered the phenomenon of CD8 T-cell exhaustion in humans. She went on to show that cytosolic nuclease TREX1, so this paper, I read it, so is a key guardian against innate immune responses to cytoplasmic DNA, enabling the virus to escape immune recognition. She and her team were the first to show that siRNA could be used to treat disease in animal models. And she developed a method to inhibit HIV sexual transmission in mice using CD4 small siRNA. She published a lot of beautiful papers highlighting the identification of microRNA that regulate different pathways. And, well, for decades she was interested in the study of siRNA, microRNA, and related methods to treat cancer. So she also studied cytotoxic T lymphocytes and NK cells and their role in immune protection from infection and cancer. And, well, she also identified something she's going to speak about, and, well, too many things. So I'm going to stop here. This was the end. And she will maybe tell you the story of what was the place of HIV in this incredible journey. So thank you very much.
It's an honor to be here. I was shocked when I was invited because I haven't really been working on HIV for the past decade, but it's very dear to my heart. And when I was an intern in medicine, there were the first HIV cases, and many people of my generation sort of switched to study HIV to try to do something about that terrible disease. But instead of what I said I was going to talk about in the abstract, I decided to talk about science because that's what I like best. And I'm going to talk about our work in innate immunity in pyroptosis and its role in infection and in HIV. And I was actually pretty surprised this morning, that there wasn't a single mention of these innate immune pathways that cause inflammation in the session on innate immunity. Because actually in the last year or so, there have been really important and beautiful papers showing that this pathway, I think, is very important for HIV. So we might as well proceed. So I thought I would start, since this is my journey, with Cold Spring Harbor. When I was a child, my uncle, Sam Granick, was the biochemist at Rockefeller, and he used to come here for summers, and actually there's a drawing of him in the room next to the dining room. And we used to go sailing, that's me over here, have cookouts, and a really nice time. And I remember, so maybe my interest in virology started here, because I remember going to a postdoc show at Cold Spring Harbor when I was probably about that age, in the late 50s, early 60s, where the postdocs sang, DNA makes RNA, and RNA makes protein, and a virus could do it too, wouldn't you? And I actually got to see, a little bit about science, when I was here and as a child, and actually biology was my first love. But anyway, let's go on to the topic at hand. So innate immunity is the way the immune system is told about what's dangerous. So we live with a lot of commensal organisms, and the immune system has to have a way to distinguish the commensal organisms that we live with that are harmless or even beneficial from the pathogens. And to do that, there are molecules either encoded by the pathogen, which are called pathogen-associated molecular patterns, or there are other molecules that are of host derivation that are in the wrong place, like DNA in the cytosol shouldn't be there, and it's a sign of danger. So these molecules, the strangers and dangers, are recognized by pattern recognition receptors. And there are two kinds of, I like to think of it as basically two broad classes of pattern recognition receptors. There are those on the outside of the cell, like the toll-like receptors, and they can't distinguish a pathogen from a commensal. And their job is basically to upregulate gene expression of molecules that are needed to respond to the pathogens, or the danger, the interest. And those are recognized. And it's a lot of the same molecules are recognized by cell surface pattern recognition receptors, as well as receptors in the cytosol of cells. And those are the receptors that can tell a pathogen from a commensal. And within the cytosol, there are a class of about almost 30 molecules that recognize these danger and infection signals. and they organize into very large complexes called inflammasomes and activate an incredibly inflammatory cell death called pyroptosis for fiery death. And pyroptosis is really the most inflammatory immune signal in the body. And it basically consists of three events. Cell death, the membrane gets damaged and forms these giant balloons. You can look at a cell and know if it's dying of this mechanism because these balloons are pathognomonic. And the cell membrane gets damaged, so the cell takes up small molecule dyes. And this pathway also activates the pro-IL-1 family of cytokines. IL-1 is the cytokine that causes fever. The whole class of IL-1 family cytokines are the most inflammatory cytokines in the body. And they get processed in this pathway and get released through these pores that form that kill the cell and damage the membrane. And at the same time, many host proteins and molecules that are inflammatory that are in the cytosol get released. They include the active forms of the inflammatory caspases that trigger this pathway, the gasdermins that poke holes in membranes, ATP, HMGB1, chemokines, and cytokines. So basically, that release of all those mediators calls forth immune cells to the site of infection or damage, and it activates them for their function. And in pyroptosis is now defined as death that's induced by a family of 5 human proteins called gasdermins. They're called that because they are expressed highly in the dermis, in the skin, and in the mucosa, like the gastrointestinal tract. And basically, any of the gasdermins can activate this fiery death, but the one that people have studied the most is called gasdermin D, and gasdermin D is in the myeloid sentinel cells of the immune system, macrophages, monocytes, dendritic cells, and also in many of the mucosal epithelia, so where the body encounters infection. But the other gasdermins, which are expressed in different locations, A is in the skin, E we hopefully have a chance to talk about is in the brain and in the placenta, places where you wouldn't think you'd want to have inflammation. So basically, pyroptosis is a double-edged sword. It can inhibit, it can protect against infection. The infections that have been studied for historical reasons are mostly bacteria. And we showed that active gasdermins can actually poke holes in bacterial membranes and kill them. So what triggers this pathway actually has the pathway actually has an immune role in defending us against. But the pyroptosis is also, in addition to recruiting cells and helping to activate lymphocytes at the site, it actually, you need a danger signal. It can be any of a variety of innate immune signals, but this is a very potent one. You need it to fully activate T cells to be functional, to kill and to have memory. So the immune system, in order to distinguish self from non-self, it needs to know that there's something dangerous to respond to. And if you have too much pyroptosis and it's a sort of feed-forward amplifying system, you can die. Too much inflammation is pathogenic. This pathway has now been shown to be critical for many important diseases, including sepsis. It's responsible for cytokine storm. But it also exacerbates diseases like atherosclerosis, neurodegenerative disease, obesity, all kinds of disease. In fact, it's actually difficult to think of an important human disease where there isn't some component of inflammation, and that includes HIV. So we got into this because, in 2000, this is a relatively new field, and you may never have heard of it, but you're not alone. Even when I give talks to immunologists, a lot of people they're not really very knowledgeable about this pathway. So in 2015, two labs, Vishwadixit and Feng Xiao, identified gasdermin D, this molecule here, as the mediator of pyroptosis in myeloid cells. And gasdermin D is composed of two domains, an N-terminal domain and a C-terminal domain, connected by a flexible linker. And that linker gets cleaved by the inflammatory caspases. Caspase 1 is in humans and mice and other mammals. Caspase 4 and 5 is in humans, and caspase 11 is in mice. And one gets activated by those inflammasomes that I was telling you about, and 4, 5, and 11 get activated by LPS, which is one of the most, powerful PAMPs from gram-negative bacteria. And it's because of this flexible loop that the gasdermins are very susceptible to proteases. And in fact, we've identified a number of proteases in the cytosol that can trigger this pathway by cleaving this gasdermin or other gasdermins. So basically, when it's cleaved, in the uncleaved form, the C-terminal domain inhibits the N-terminal domain. When it's cut, they separate, and the N-terminal domain forms pores in membranes. So we showed that this molecule forms pores in membranes, and that's how it works. Because Xing Liu, a postdoc in my lab, wanted to try to figure out what the gasdermins were doing. And at that time, when they were discovered to be the mediators of this pathway, nobody knew. And there was nothing known about the gasdermins. There were maybe 5 papers in the literature. And Xing fed the N-terminal domain into a homology search program. And there was only homology to these other gasdermins, and nothing was known about them. But there was structural homology predictive of this N-terminal fragment to perforin, the pore-forming protein in killer lymphocytes that my lab had studied a lot. So I said, it must be a pore-forming protein. Let's figure it out. And so the first thing we did was try to figure out what lipids the N-terminal fragment of gasdermin D might bind to. And we used lipid strips that were dotted with different lipids. And it turned out that the N-terminus, but not the full length or the C-terminus, bound to acidic phospholipids that are only on the inner leaflet of the cell membrane. So this is a pathway that works from within. The gasdermin gets cleaved in the cytosol, and it forms pores on the inner aspect of the cell membrane to permeabilize it. But it also bound to cardiolipin, which is on mitochondrial and bacterial membranes, and that's going to turn out to be important. That's the mechanism by which gasdermins kill bacteria, by binding to cardiolipin. And the only bacterium that we could ever find that was resistant to this pathway is a mutant bacterium that lacks cardiolipin synthase. But mitochondria are sort of like bacteria, ancient bacteria that live in our cells. And it turns out that this binding to cardiolipin is important in the cell death pathway because gasdermin binds about 10 times more tightly to cardiolipin than to these other phospholipids. So it actually goes, once it's activated, it goes to the mitochondria, blows them apart, and that sort of is essential for the cell death pathway. And then we were able to show that if we added full-length gasdermin D to lipid membranes, nothing happened. If we added one of these inflammatory caspases here, caspase 11, nothing happened. But together, we could detect pores on the surface of the cells undergoing pyroptosis. And they're very large, several hundred angstroms in size. And a lot of my work on pyroptosis is a collaboration with Hao Wu, and Chinbin is in her lab. And Hao's lab, she's an amazing structural biologist, solved the structure of this pore. This is the inner side, the cytoplasmic side, and then it's like a giant beta barrels with those beta fingers sort of digging into the membrane. And it's it's an intelligent design of a pore because the inner rim in the cell is acidic. It's incredibly acidic. The pro-IL-1 family cytokines are acidic on their surface. They're repelled. But once they're cleaved and activated, they have a basic surface and they get through. Okay, so one of the places where this pathway is incredibly important is in sepsis, which is really an important killer. Basically, it's the most important cause of cell death in children throughout the world, and a third of all hospitalized patients suffer from complications of sepsis. And there's no treatment other than supportive care, even though there have been hundreds of trials to try to intervene and prevent it. But sepsis due to LPS and probably other gram-positive bacteria as well is dependent on this pathway. So in mice, if you inject a lethal dose of LPS into wild-type mice, they'll die within a day. But if they're knocked out for the inflammatory caspase 11, which actually senses LPS, most of the mice die, or if they're knocked out, survive, or if they're knocked out for gasdermin D. So basically, the gasdermins are like a bottleneck for this pathway. There are many sensors of DAMPs and PAMPs, both on the cell surface and inside cells. There are here 27 inflammasome proteins, and plus these inflammatory caspases. And actually what many of them recognize, we still don't know. But they recognize a variety of things like toxins and bacterial flagellar and needle proteins get delivered during from type 3 secretion systems of bacteria. When they recognize what they recognize, the inflammasome forms, and it's like a micron. It's a huge machine, micron in diameter, that recruits the proform of caspase 1, one of the inflammatory caspases. It gets activated by autoproteolysis in proximity, and it both cleaves these IL-1 family cytokines as well as gasdermin D, which forms pores that enable these active proteins and other molecules to be released. And that's called the canonical inflammasome. And then there's a non-canonical inflammasome, which involves, it's a simpler system. LPS binds to caspase 4 and 5, activates it, and it cleaves gasdermin D. And this pathway doesn't process the IL-1 family cytokines, but when the membrane gets damaged, one of the NOD-like receptors, NLRP3, which you may have heard of, people have indicated it might be involved in HIV inflammation, gets activated by recognizing some feature of membrane damage, and so this pathway also gets activated at the same time. So we thought, since gasdermin D is like a bottleneck for this process, if we could develop an inhibitor or identify an inhibitor of gasdermin D, we might be able to block sepsis or other inflammatory pathways. And so Junhoo in Hao's lab and Jing in my lab did a high-throughput screen in which we took liposomes made up of lipids that are that gasdermin D recognizes, loaded them with terbium, and then incubated them with caspase 11 and full-length gasdermin. And we were looking for molecules that would block liposome leakage, which we could measure because the medium had this dipicolinic acid that reacts with terbium to become fluorescent. And so we identified only one molecule that actually worked in cells as well as in liposomes, and it's actually an approved drug used to treat alcoholism called disulfiram or antabuse. And it's a cysteine reactive drug. It was active at nanomolar concentrations, and it bound to a cysteine, which we had identified in our first paper as critical for pyroptosis. And it's actually just at the tip of all those fingers eating into the cell membrane, so it's accessible. And if we treated mice that were challenged with LPS, we could partially block, here's disulfiram in black, partially block death from sepsis, but also have a profound effect on inflammation. Here we're measuring serum cytokines like IL-1. It went down substantially, TNF and IL-6. And around the same time, Kate Fitzgerald identified another cysteine reactive drug that also binds to the same site. And that's a drug that's used to treat multiple sclerosis as another inhibitor that blocks sepsis. Okay, so we had a drug, but we couldn't get anyone to, develop it because it's, first of all, drug companies don't like cysteine reactive drugs because they have off-target effects, but also it was been used for 70 years, so there's no patent protection. But anyway, so I just wanted to show you the data about gasdermin D killing bacteria. Here we just took E. coli and Staph aureus, gram-negative and gram-positive, incubated it with nanomolar concentrations of gasdermin, the N-terminus, or a catalytic pore mutation that doesn't form pores, or the C-terminus or the full length. And you can see that we get death of bacteria by colony counts. This death is incredibly fast. If you treat bacteria with antibiotics, It takes several hours to kill bacteria. This happens instantaneously in minutes. And it works by binding to cardiolipin. So because we found that bacteria are killed, we thought, well, most apoptosis, mitochondrial damage, is a key feature of that cell death pathway, which Doug Green once called the point of no return, that it's really critical for executing apoptosis. So we wanted to see if mitochondria might be targets of pyroptosis because they had the right lipid. And we also wanted to know, would mitochondrial damage be important for killing cells? And so this work was done by Ray, Winston, and Jing in my lab. And basically, in classical apoptosis, the outer membrane gets permeabilized, releasing things like cytochrome C from the intermembrane space. But in pyroptosis, which actually occurs very quickly within minutes, as soon as we can detect gasdermin deactivation in cells, it damages both membranes and releases contents both from the intermembrane space as well as the matrix, including DNA, which is incredibly inflammatory on its own. So this is just the data where you can see activation of caspase 1. The first time point here is 7.5 minutes. At the same time, we begin to see cleavage of gasdermin D. And then we start seeing processing of IL-1 beta. So in red is the cytosol, in green is the proteins in the mitochondria, and in blue, proteins that are secreted into the medium. So you can see that the release of cytochrome C begins to be detected within about 10 minutes. From mitochondria, it appears in the cytosol, and there's a delay in the damage to the cell membrane because the release only occurs at about a half hour. So this process starts in the mitochondria, it blows them apart, it destroys mitochondrial metabolism, it induces reactive oxygen species, and it releases soluble proteins from either the intermembrane space or from the matrix, like aconitase, not membrane-bound proteins like COX-4, and it also, beginning at 7.5 minutes, releases mitochondrial DNA into the cytosol, which is immunostimulatory. And this mitochondrial damage is critical for killing the cells. So here we took advantage of ethidium bromide to delete cells of mitochondria, here stained with mitotracker in the untreated cells, and compared cell death after stimulation with LPS and nigiricin, which activates the NLRP3 inflammasome. So in untreated cells, there's 60% of the cells die, but only less than half die in ethidium bromide treated cells. And for the LPS reaction to intracellular transfected LPS, the difference was even greater. And we showed that this death depends on oxidative ROS. If we use ROS scavengers, we inhibited death, and if we treated cells with oxidants, we increased it. And this cell death depends on cardiolipin. So cardiolipin is first synthesized on the inner membrane of mitochondria, and then there's a scramblase called PLSCR3, which takes it from the inner membrane to the outer membrane and flips it to the outer leaflet of the outer membrane. And if we knock out the synthetase, we get a dramatic reduction in cell death in cells stimulated with salmonella. If we knock out the scramblase, the same thing. And those knockout cells don't secrete any IL-1. I don't know why there's some cell death here, but no release of IL-1, but that's what we found. And so in two recent papers that I was involved in, but they were done in other labs, we found that this cysteine that disulfiram responds to is critical for pyroptosis because it is lipidated on this cysteine. A palmitate is transferred to the cysteine, and that lipidation helps gasdermin go to the membrane, embed in the membrane, and form pores. So if we take mammalian gasdermin D and do this liposome leakage assay, we get a lot of cell death. If we remove the palmitate from the mammalian cells, there's very little cell death. If we use the cysteine mutant, there's very little cell death. And if we use bacterial recombinant gasdermin D, there's not very much cell death either because the bacteria don't have the palmityl transferase. And here you can just see, if we look at gasdermin D and propydium iodide, which gets into cells with damaged membranes, you can see that if we treat with LPS and nigiricin, we get these giant bubbles and pyroptosis. The dye goes into the cell. But in cells that express the mutant with the cysteine, that's alanine, there's no pyroptosis. And this palmitylation depends on the ROS that's generated in this mitochondrial pathway because, so here we're staining the cells the same. We have a GFP labeled gasdermin and propydium iodide dye. So the control, we see pyroptotic bubbles, the dye entering. If we inhibit the palmityl transferase with bromopalmitate, there's no pyroptosis. If we add oxidants that generate ROS to the cells, like antimycin A or rotenone, we get pyroptosis that's blocked by blocking palmitylation. And if we scavenge ROS with these ROS scavengers, there's also no so this sort of links the two stories I told you, that this cysteine has to be palmitated, and it's palmitated only in the presence of reactive oxygen species. And so this is actually the first example of regulation of this pathway. It's really a checkpoint. If we block palmitylation, then this gasdermin is inactive. And these are the people who really did most of the work on these two papers. So now I'd like to go over to COVID, another virus. Okay, so the reason I got interested in COVID was, first of all, severe COVID looks a lot like sepsis, and cytokine storm is what's important in severe COVID. There are many inflammatory pathways in the cell, but this is actually the most inflammatory. So there was reason to believe it might be involved in COVID. So we wanted to know, was it? And if so, how was it induced and in what cells? And these are the people who led the study in my lab: Caroline, Angela, and Shaheen. Basically, we started out by looking at the fresh blood from people who were seen in the emergency room of MGH for respiratory symptoms. And we had to use fresh blood because if you freeze lymphocytes or any cells that are dying, when you thaw them, they die and you don't recover them. So we looked at fresh blood. We stained with a dye that's fixable, so we could use it for SARS, zombie yellow, and for an Exon 5 that marks classical apoptotic cells. And what we found is a very subtle increase in zombie yellow staining in monocytes, about 6% in this patient, but no change in cell death, either apoptotic cell death or pyroptotic cell death in lymphocyte populations. And here we're looking at healthy donors on top and COVID patients on the bottom. And when we also looked in the plasma of patients who went on to who were either healthy donors or went on to develop mild disease, moderate disease, or severe disease that required ICU. And what we found, and we looked at markers that are specific for pyroptosis, namely gasdermin D in the blood, LDH, which gets released from cells dying of necrosis, IL-1, the IL-1 receptor antagonist, or IL-18, another IL-1 family member. What you can see is the levels are up in COVID compared to the healthy donors, and they're more up in people who develop severe disease. And then we use a technique called imaging flow cytometry, which is a combination of microscopy and flow cytometry. So you get images of tens of thousands or hundreds of thousands of cells. And we stain either healthy donor or COVID patients for the adapter for the inflammasome called ASC, which is necessary for assembling many of the inflammasomes, for gasdermin D, which is normally cytosolic. And what we found is these large specks in some of the cells from the COVID patients, and also the gasdermin D redistributed to puncta on the cell membrane. And then when we stained the same healthy donors or COVID patients with the dye, the zombie dye and gasdermin, what you can see is the morphology is these giant balloons. The gasdermin is actually permeabilizing the membrane at those balloons, and the dye is getting in. So patient monocytes were dying of pyroptosis, but they're not supposed to be infected because they don't have ACE2, the receptor. And it turned out that we found that these inflammasomes activated NLRP3, AIM2, which is a sensor of cytosolic DNA, but not pyrin, which is a sensor for bacterial toxins. And the same inflammasome spec would have all of these inflammasomes. There might be other inflammasomes involved. Mostly there aren't any antibodies for most of the inflammasomes. And so what activates inflammasomes? We thought, well, maybe the monocytes are infected. So we stained for nucleocapsid or with an antibody that recognizes double-stranded RNA, which only is produced when the virus starts to replicate. And we found that there was basically a one-to-one correspondence between cells that had ASC specs and stained for nucleocapsid or for J2. So basically, somehow, these monocytes were taking up the virus. Maybe they were beginning to replicate it because otherwise they shouldn't have double-stranded RNA in the cytosol. And that uptake was triggering these aspects. So basically, for example, if we look at the cells that stained for double-stranded RNA and looked at whether the cells that didn't had aspects or that did, you can see there's a pretty good correlation between ASC positivity and J2 positivity. Similarly for I'm sorry, I'm lost here. But anyway, basically the cells that had aspects were infected and the cells that were infected had aspects. And then we looked in the lung of patients these were autopsies of either people who died of traffic accidents or COVID. And we stained for aspects for nucleocapsid in green and for CD14, which is the marker of macrophages. We also stained for epithelial cells and endothelial cells. But basically what we found is that aspects were in the lung were only in macrophages, but they were not in the CD14 negative endothelial and epithelial cells. And then when we quantified how many cells were undergoing pyroptosis that had aspects, among the macrophages that were CD14 positive, there was no background in the control samples, but only about 5% of the macrophages in the lung were infected, but a quarter of all the macrophages in the lung were undergoing pyroptosis. That's huge. And in the blood, about 5% of the circulating monocytes were also undergoing pyroptosis. So now you might see why you get cytokine storm with this infection. And in the epithelial and endothelial cells, although about 15% were infected, which isn't that much more than the rate of infection of the macrophages, that none of them had aspects. And we don't really understand why the cells that get infected through the receptor, the normal receptor, don't trigger pyroptosis, but the cells, the macrophages that don't have that receptor do. So we wanted to know, do monocytes and macrophages actually get infected? Do they replicate the virus? Do they produce infectious virions? And we noticed that, so in the blood, monocytes are classified according to two receptors, CD14 and CD16, which is an Fc gamma receptor. And most of the monocytes in healthy donors, in white here, are classical monocytes that have high expression of CD14 and no expression of CD16. In COVID patients, there's somewhat fewer of these classical monocytes, and there's an increase in monocytes that express CD16. And then when we looked in the imaging flow cytometer, we found that only the CD16 positive monocytes became infected in patients, had nucleocapsid staining, but the CD16 negative ones didn't. And actually about half of those non-classical populations became infected. So then we wanted to verify that this was the case. We took healthy donor monocytes and incubated them with SARS virus, and then we used the molecular clone that has a neon green reporter and stained for detected neon green and nucleocapsid and ASC specs. And we found again that the cells that were infected had ASC specs, the uninfected cells didn't, and that infection required the presence of either patient plasma or antibodies that recognize the spike protein, but they didn't have to be neutralized. If any antibody worked. And if we depleted the plasma of IG, then we got much reduced infection. And again, in this setting, the cells with aspects were all infected. And so we thought that this receptor, the infection required antibody coding. It seemed to require this receptor CD16. So what would happen if we did the infection in the presence of antibodies to CD16, another FC receptor on monocytes that's very widely expressed, ACE2, or another receptor which was claimed to be involved in entry, or depleted the IG. And what you can see that is that from the plasma, that blocking CD16 strongly reduced infection, or depleting the plasma of IG also. So it seemed like this infection was mediated by antibody uptake by an FC receptor. And then we wanted to know what about viral replication. So I told you that we could stain for J2, suggesting that viral replication had begun. But we wanted to confirm that, so we looked at the subgenomic RNAs that are transcribed during infection, and we found that in the uninfected monocytes we couldn't detect these subgenomic RNAs, but in the COVID-infected healthy donor monocytes we did. So the virus started to replicate, but we couldn't isolate any infectious virus 2 days later. So basically, what we think is going on is this is a very, very fast cell death pathway. It takes about 12 hours to replicate this virus. The virus comes in, it triggers pyroptosis, and it never gets to complete its life cycle and produce infectious virions. So this is a pathway that is partly protective. You don't produce infectious virions, but it creates a hell of an inflammatory environment that can kill the patient. And the importance of this pathway was tested by Essen and Richard Flavel's lab, which we collaborated with. And they have a mouse called the Mr. G mouse that also expresses the human ACE2 receptor. It's a humanized mouse model. And the Mr. G are encode for cytokines that are important in the development of innate immune cells like myeloid cells and NK cells. So it has a really full immune system. And what Essen found was that in the serum of these mice, when they were infected with SARS, they became infected and they produced in their serum very high levels of IL-18, IL-1 receptor, and human gasdermin D. These are all human, not mouse. And we were able to do this imaging flow cytometry analysis of the human hematopoietic cells in mice that were either infected or not, and found that aspects, NLRP3 inflammasomes, were present in the infected human monocytes in this model. And importantly, they were able to use this model to see if this pathway was important in lung pathology. So they reconstituted the mice, gave them through an AAV vector the human ACE2, infected them with SARS after they showed they were engrafted, and then treated them with either inhibitor of caspase-1, which assembles the canonical inflammasome, or an NLRP3 inhibitor, and looked in the lungs. And what you can see is the pathology, the inflammation and fluid in the lung is much higher in the control mice than in the mice treated with either of those inhibitors of this pathway. So basically, this is an example, but I want to tell you an HIV story. So I'm going to go through. I'm not going to summarize what I just told you, but basically, in severe COVID, there's activation of this inflammasome pathway in a large number of macrophages, tissue macrophages, and monocytes, and we believe that's important in driving the pathology. Okay, so let me just switch to HIV because in the last year, there were 2 papers that came out about an inflammasome called CARD8, and that it's important in HIV infection. And I want to just show you some of the data because, so Warner Green, I don't know, 10 years or so ago, what? More than that? Really? Anyway, he found caspase-1 activation and IL-1 in SARS patients, and he thought it was responsible for CD4 depletion in HIV. I know a number of labs tried to reproduce some of his findings, including ours, and we couldn't, but maybe it was a very complicated experimental system, and that might have been why. But anyway, these papers actually implicate another pathway of inflammasome activation by HIV, which I think is responsible for inflammation and CD4 depletion. And it involves an inflammasome receptor called CARD8, and it's a very complicated inflammasome. This is its structure. It has what's called the fine domain, which is basically a domain that nobody knew the function of. The protein gets auto-processed by cleavage here, not known how that happens. But the 2 halves stay together. But then at the N-terminus of the protein, there's a disordered region. And disordered regions are very susceptible to proteases. And they're actually, in this case, a sensor for proteases. And what has been shown is that the HIV protease cleaves here. And once it gets cleaved, the N-terminus of this fragment is susceptible to ubiquitination and protein degradation. This part gets degraded and releases, this is the proteasome, and releases this fragment, which has a CARD domain. And CARD domains are important in nucleating inflammasomes because they're homotypic interaction domains. So it recruits caspase-1, activates it, and causes pyroptosis and secretion of inflammatory cytokines. And I'm going to show you some of the data. So basically, what this paper in Cell in February is beautiful. I really recommend it to people, even if you're not at all interested in immunology. It's beautiful, and I think it's important. And basically, what they found is that pyroptosis was activated in a virus-dependent manner, but if you blocked the protease with LPV, there was no inflammation. And here they used, if you inhibited the entry of the virus, you inhibited pyroptosis or the protease, but integrase or reverse transcriptase inhibitors had no effect. And if you knocked out either CARD8, caspase-1, or NLRP, or ASC, no, not ASC. This inflammasome doesn't require ASC because it has a CARD domain. Or if you knocked out gasdermin D, you blocked the cell death from HIV. So basically, if you inhibited the protease, knocked out CARD8, knocked out caspase-1, or knocked out the pore-forming protein, you didn't get pyroptosis. And what they found is that this pyroptosis only occurred in resting CD4 T cell and not in activated CD4 T cells. And why that is, no clue. And if they made a humanized mouse in an immunodeficient background and knocked out CARD8 in the hematopoietic stem cells, they found CARD8 activation with HIV infection. So here's the fragment, but not in the knockout mice, that the viral load was higher in the knockout mouse, and the CD4 count was also higher in the knockout mouse. So basically, I think that's pretty good evidence in vivo, at least, that this pathway is reducing viral production, although they never showed it in the paper, and reducing CD4 depletion, and it only works in resting cells. So the cells have to be, the virus has to enter the cells, the protease has to be active in the cells in order to trigger this pathway, but it doesn't have to go on to reverse transcription. And the other thing that this paper showed, which is really elegant, is that the CARD8 protein in humans, which has this CARD domain, which is critical for forming the inflammasome, in the monkeys that that live with HIV without symptoms, they do not have a full CARD domain, so they can't activate this pathway. Whereas the monkeys for which SIV is pathogenic have a full CARD domain. And I'm going to skip all the data, but basically the CARD domain, if you took CARD knockout cells and reconstituted them with different either susceptible animal CARDs or asymptomatic animals like pseudomangabes, only the susceptible ones developed pyroptosis, processed IL-1, and caused rapid CD4 depletion. So that in this case, they co-cultured resting CD4 cells with infected SIV MAC251 infected cells and measured the CD4 to CD8 ratio. And with the pseudomangabee cells, there was no CD4 depletion, but with the rhesus macaque CARD8, there was a drop from about 50% to 35% in CD4. And this was only within 16 hours. Okay, so basically, cardate is a cytosolic sensor. It has this floppy domain and the N-terminus that's susceptible to proteases, including the HIV protease. It cleaves it early after entry. I believe pyroptosis is probably activated really fast before the virus can continue its life cycle and produce infectious virions. It involves this canonical pyroptotic pathway with caspase-1 and gasdermin D. There's evidence that it's only active in resting CD4 cells. We don't know why. And it's only active in the non-human primates who develop SARS disease. So again, this pathway is a double-edged sword. It reduces viral load by, little less than a log, but a lot. But it increases chronic inflammation, and that inflammation is responsible for the accelerated aging and many of the side effects of people who are, infected with HIV, even who have viral control. And basically, in my mind, this pathway actually explains some of the things that I wondered about when I was working in HIV. Why is there CD4 T cell depletion when only 1 in a million cells or so become infected at any one time? Because it's the resting cells that actually are susceptible. What is the mechanism behind the chronic inflammation in HIV and its long-term side effects? And why are some primates susceptible and others not to SIV disease? And so I'm not going to give the rest of my talk because I went over, but there's also another HIV problem which I think is related to pyroptosis, namely the neurocognitive brain disorder, but that it likely involves gasdermin E, another gasdermin. I don't have time to tell you about it. And these are the people who did the work. So I'd be happy to answer any questions. I didn't do the HIV work, so I might not know the answer to questions about it.
So much beautiful work. With gasdermins, do they show any signs of positive selection, suggesting viruses have tried to get rid of them?
Well, actually, there's very little work on virus infection and this pathway, so I don't know the answer to your question. But like cancer cells suppress the expression of gasdermin E, and it helps them evade immunity. Mm-hmm. And there are mutations that are linked to cancer, but nothing about. I mean, I think if I were a virologist, this is a really important pathway. You have a sensor that detects proteases in the cytosol, and that paper is very convincing to me. Yeah.
Amazing talk. I was thinking about the connection between gasdermins and pyroptosis, but also the idea that like senescent cells secrete a lot of IL-1 beta, and they seem to not die. Do you believe in life after inflammasome activation, and how does that occur?
Yeah, okay, so yes, there is life after inflammasome activation. So when I was working on perforin, I showed that perforin, in order for killer cells to work and induce apoptosis, they had to repair the membrane damage that they caused. And so there's every cell has a membrane repair defense because membranes are damaged all the time. So in some cells, especially in myeloid cells, the repair of the cell membrane can be such that the cells don't die, but they're so inflammatory. So if they don't die, then they keep churning out more and more inflammatory cytokines, like 10 times as much as the cells that die. So and in fact, one of my colleagues, John Kagan, has shown that those myelodendritic cells that undergo what he called hyperactivation, so they activate pyroptosis but don't die, are the most potent antigen-presenting cells that you can imagine, more potent than pyroptotic or apoptotic dendritic cells.
Hi, that was a great back here. That was a great keynote. I really enjoyed hearing about the scientific journey. I was actually kind of really interested in the SARS aspect of your talk. You said that the FC positive cells were the ones that were infected, but that was only with serum from patients or with spike antibody. Have you done any experiments to separate out FC-mediated phagocytosis versus active infection? Because if it's circumventing the ACE2 pathway, it's not getting into a acidic compartment to properly release the RNA.
No, it's getting to it's being endocytosed, so it is an acidic compartment. I think our data suggests that viral replication does start. We see double-stranded RNA. We see these intermediates that occur later in the viral life cycle. But there is something very different about the virus uptake by ACE2 and by the FC receptor, because if we transfect in ACE2 into THP1 cells, they get infected, but they don't turn on this pyroptotic pathway, and they have all the machinery. I also want to make another comment. So when we found this, I was very concerned that, what about vaccines? Because they're producing antibodies and maybe they're harmful. But what we found is that the CD16 is very sensitive to antibodies that are afucosylated, that don't have a fucose on their constant region. Like they bind to afucosylated antibodies about 10 times more strongly than to fucosylated antibodies. And it turns out that the patients who had been vaccinated with the Pfizer vaccine, who we tested and had antibody titers as high or higher than the people who appeared in the ER with acute COVID, their plasma did not activate infection. So that was reassuring. But we have to study that more than we have. And I make one other comment because it's really interesting. We have also been looking at long COVID patients, and we have found, this is very preliminary, but we found that if we isolate immune complexes from the blood of long COVID patients, they have their immune complexes stimulate pyroptosis. I don't know the mechanism. And they have cells in their circulation that are undergoing pyroptosis, and not in the convalescent patients who don't have long COVID. So I think maybe there's some connection there, but it's a work in progress.
Yes, thanks a lot. I was really impressed. So my question is also about the SARS part. Some people speculate that Omicron is attenuated also in people. So do you think that actually SARS-CoV-2 may evolve towards reduced pathogenicity? And did you compare the early and the late strains of SARS-CoV-2?
We did. We didn't find the difference in this pathway but Jeremy probably knows a lot more about it than I do.
So I was curious, thinking back to Warner's story, his story revolved around incomplete reverse transcription, aborted transcript, activating caspase-1 somehow, and he never invoked the protease.
Right. So his was, he said that the sensor was IFI16, which is a DNA binding protein. But I don't know if anyone has ever verified. I think it's possible that I couldn't, and we spend a lot of time trying to.
So in SARS-2, in the SARS-CoV-2, have you looked in more detail at what the products are? So you've shown there's double-stranded RNA, so there's some replication going on. But is it complete, or is it just the short?
All that we've done is in that one slide. We haven't looked in more detail, but
and there's no
think,
evidence.
As I said, this cell death is like so fast. It's like, but the truth is, if you look at the literature about SARS, even with the immense research that has been going on in the last 5 years, there is not that much virology there about, really understanding how the virus replicates, how long it takes, it's like really shocking. And we would like to understand better how, what about SARS is triggering this inflammasome response. But the problem is that because of, we only have a small number of cells that get infected, so it's very hard to work with. So we've been trying to enrich for the CD16 positive cells or develop THP1 cells that we can infect through this pathway, which we haven't yet.
Would any of the SARS-2 proteases touch caspase-8?
It's possible, or they might touch cardate. Yeah. I think that's, when I was preparing my talk and rereading these papers, I said, why didn't we look at this? But there are other viruses, but I don't remember which where that viral protease is implicated in activating. And there are bacterial proteases that in mice, it's NLRP1 is the homolog of cardate that have been implicated in the cleavage at the N-terminus.
Thank you. In acute COVID, in severe acute COVID, there is a very rapid CD4 depletion.
A very rapid what?
CD4 T cell depletion.
We didn't look at all at that.
So
we
I
didn't
wonder
see
if there
any.
is some pyroptosis involved.
We didn't see any evidence for so some lymphocytes have CD16 receptor, and neutrophils have CD16. We didn't find any evidence for pyroptosis in either of those or in NK cells have CD16. So, and we didn't look at we didn't see any evidence for pyroptosis in those cells. We looked.
So maybe we are going to have our last question over there.
Yeah. So looking at the Gas-German pore, it's reminiscent to bacterial pore-forming toxins, pneumolysin pores like this. So my question is, can these bacterial pore-forming lysins also trigger pyroptosis? Or
they should.
Something similar?
Yeah, so if the cells, not all cells have NLRP3, but NLRP3 is the sensor for membrane damage. So anytime your cytolysins, listerolysins, all these things, they will form pores in the cell membrane that should trigger pyroptosis if the cell expresses NLRP3 and caspase-1. So all of these molecules, there's not a lot known even about what cells express what, because even if you look in the protein atlas, a lot of these genes that are involved in these pathways are induced by interferon gamma, other inflammatory cytokines. So, but you have to look if the target. So, like for CTL killing, I always thought of that as apoptosis, non-inflammatory, no bystander effect. But what we found is that granzyme B actually cuts gasdermin E and triggers pyroptosis if the cell happens to express gasdermin E. So you have to know what the cell's expressing to predict what pathway might be activated. But definitely there are going to be cases where those bacterial toxins, pore-forming proteins, cause pyroptosis. And actually we discovered the only known activator of the gasdermin in the skin, gasdermin A, is a virulence factor of group A strep that cuts gasdermin A, which we discovered. But so this is like a new field. There's a lot that can be done in it.
Thank you very much. I hope we can continue the discussion during the poster session. I hope you are full of energy to enjoy the first poster session. Yeah.